Hey everyone, Krista here. Today we're going to take a little trip around a eukaryotic cell and talk about the major organelles. So since we're talking about something microscopic, a cell, I like to give my students a visual to think about. So if you were to imagine a balloon, and within that balloon, we're going to stick some pudding. And then within that, we're going to drop some jelly beans. So the rubber of the balloon would represent the cell membrane. The pudding inside the balloon would be the cytoplasm. And then those little jelly beans are our organelles. And so when we're talking about our organelles, think of the word organelle. So this literally means tiny organ. Their whole purpose is the division of labor. So this really helps eukaryotic cells be more complex. And so when we're talking about different parts of the organelle, one of the big things is the plasma membrane, right? So this is what encompasses the cell, the outside, that rubber of the balloon. And so in biology, we use many terms interchangeably. So plasma membrane, cell membrane, also phospholipid bilayer are all the same thing. And the reason a cell membrane or a plasma membrane is also referred to as a phospholipid bilayer is because we have two layers of phospholipids, so bi meaning two. So pictured here, we have those little yellowish orange heads, those little balls, and then we have two white lines underneath that. So the head of the phospholipid is hydrophilic. So hydro meaning water, philic meaning to love or like. So they like to be around water. The outside of the cell is aqueous. The inside of the cell is aqueous. So those phospholipid heads are going to be pointed out towards the outside of the cell and then pointed in towards the inside of the cell where all the aqueous solution is. And then the phospholipid tails are hydrophobic. So hydro, again, water, phobic means fear. So they do not like water. So they're going to fold towards the inside of that bilayer and be protected from the aqueous solution. The next organelle is the flagellum. So the flagellum is an appendage used for locomotion made of microtubules. Not all eukaryotic cells are going to have a flagellum, but when they do, it's used for movement. And so the most notable one that we all think of is the sperm cell. They have very long flagellums. The next organelle is the smooth endoplasmic reticulum, also referred to as the smooth ER. So the smooth ER is involved in the manufacture of lipids. Lipids are fats, oils, all that stuff. So these are hydrocarbon chains. And we'll talk about that in a subsequent lesson. But for now, one of the best ways that I use to remember what the smooth ER does is, these manufacture lipids or oils. And if we wanted our, let's say, hands to be smooth, we could rub an oil on it. The next organelle is the rough ER, so the rough endoplasmic reticulum. This is involved in the manufacture of proteins. And again, my silly way to remember this is if you're rough and tough, maybe you're eating a lot of protein. So here it's pictured in yellow, and you can see those little blue dots on it. Those little blue dots are ribosomes. And again, in a subsequent lesson, we'll talk about this in more detail, but ribosomes are very involved in the manufacture of proteins, and they actually help link up those amino acids, those small little subunits that make up proteins. The next organelle, here it's pictured in green. And when you're looking at these illustrations, the artist that makes them is going to use all different colors. So they could definitely vary in color, but you're going to see this organelle that looks like kind of like stacks of pancakes. And so the Golgi apparatus, or the Golgi vesicles, usually we call it the Golgi apparatus actually, is mainly involved in telling manufactured components, anything within the cell, where to go. So when the cell is making something, sometimes those things are going to be kept within the cell, and sometimes they need to be exported out of the cell. And so you can kind of think of the Golgi apparatus as the post office. It's going to put a chemical signature on that substance and basically tell it where to go. So then we're looking at the nuclear complex, that I like to call it, or just the nucleus. But the nucleus has many, many parts. And so the first part here is the nuclear pores. So a pore is just an opening. Okay, and so this really allows exchange between the nucleus and the cytoplasm. Next, we have chromatin. So chromatin is a complex of DNA and proteins. Outside of that, we have the nuclear envelope. So this would be almost similar to our plasma membrane. It's just the membrane that goes around the nucleus. So it envelops the nucleus. And then we have the nucleolus. So that is the very, very center of the nucleus. And this is involved in ribosomal biogenesis. So basically making ribosomes, making RNA. So next here, we have this little blue ball that is a peroxisome. Again, this could be depicted as multiple colors in various illustrations. Here, it's depicted in blue. And so peroxisomes contain enzymes that move around hydrogens. One of the biggest things to remember about it is involved in making and then degrading hydrogen peroxide, so hence peroxisomes. And then you'll see that little orange noodle-looking thing. This is a microtubule. So we have multiple protein filaments that make up what is called the cytoskeleton of the cell. So going back to our analogy of we have that balloon filled with pudding and the jelly beans, but now picture that we don't want the balloon to kind of lay flat and just be a blob. We need it to have some structure. So let's say we're going to put some uncooked spaghetti within that pudding mixture, and we're just going to make it so that it actually has like a cylindrical shape, right? So that it's not just flattened. That is our cytoskeleton. And our cytoskeleton is also the way that things are going to move around the actual cell. And so the cytoskeleton is made of multiple types of filaments that are protein-based, and the largest of those are these microtubules that are actually like tubes. And next we have lysosomes. So here it's a little difficult to see, but it's kind of that globular, orangish, yellowish thing located right behind the mitochondria, which we're going to talk about in a second. But lysosomes are used to break things down. They have digestive enzymes. And so this is really important when we are dealing with waste inside the cell. And so lys, L-Y-S, means to break. And I will say, any of you who are watching this who are thinking about, being a science major or going into medicine, I would very highly recommend learning your Latin roots. This will help you tremendously. But anyways, lys, L-Y-S, means to break. So lysosomes break things down. And then we also see that we have some free ribosomes. So this is just to note that we're going to have ribosomes that are located on our RFER, but we are also going to have free-floating ribosomes. Again, these are involved in the manufacture of proteins, and more specifically, they help with actually matching up the correct amino acids, those subunits, to manufacture specific proteins. The next organelle we have here, and as you can see, we've got two arrows because here there are two depicted, is the mitochondrion. So one of the best ways to be able to pick out the mitochondria are, you're going to see that little swervy line in there. And what that's actually depicting is that the mitochondria itself has a double membrane. When we go into our lesson later on the mitochondria, we're going to see that that double membrane is really, really important when we're manufacturing something called ATP. So that's adenosine triphosphate. So ATP is our energy. This is actually how we manufacture energy within us, and how all eukaryotic cells manufacture energy. And so those double membranes come in very, very handy. But the biggest thing to know, let's say if you were being tested on this, is that this is like the powerhouse of the cell. It's producing ATP. And then we have the intermediate filaments. And so the intermediate filaments, here it's depicted as kind of a light green little spaghetti looking thing. These are intermediate, so they are the middle-sized filament that makes up the cytoskeleton. And then we have the cytoplasm. And I put that graphic on there just to show you, The cytoplasm, again, it's aqueous, it's gel-like, and it's all of the space pretty much inside the cell that all these organelles and the cytoskeleton are suspended within. And then we have secretory vesicles. And so these are whether or not a cell wants to take something in or expel something, it's going to move around the cell in what is called a vesicle. So I always think like vesicle, vehicle. And so oftentimes when we need to expel something outside of the cell, we're going to call it a secretory vesicle, because what has happened is that it has moved through the cell and it's going to travel to the plasma membrane. It's going to actually fuse with the plasma membrane and expel whatever it needs to expel. And then we have our centrosome with our two centrioles. So these are mainly used in cell division, and they are made of microtubules, and the centrosomes all have two centriole pairs. So that is, you know, a good synopsis of a eukaryotic cell, but an animal eukaryotic cell. So remember, both animals and plants are eukaryotic cells. And so let's just take a look at the differences. So a plant cell is going to have everything that we just talked about, but some extra stuff. Plants are super cool. So the first big difference is the cell wall. So they have another layer outside of the cell. So here in this picture, that yellow, that little sliver of yellow on the inside, that's our plasma membrane, just like us, just like animal cells. But then outside of that, there is going to be a very rigid cell wall. And next we have a central vacuole. Now, animal cells also have vacuoles, but ours are super, super small. All plant cells have a very large central vacuole. And these have different metabolites and chemicals in it, but really it's just kind of like a big lake, a big water reserve. And so we're going to learn about how plant cells go through photosynthesis, and we all know plants need water, and more specifically, they have to have it for photosynthesis. So it's really good to keep a very large water reserve on hand. And also it's going to be involved in something called turgor pressure. So this is a very important pressure for cells to maintain, for plant cells to maintain, to be able to move things. And then most notably in the plant cell, in any photosynthetic organism, we are going to have chloroplast. So this is where photosynthesis is taking place. So this is how all photosynthetic organisms are harnessing the energy of the sun to make actual food energy. And so that is basically it as far as our eukaryotic cells go. So I did skip over some of the lesser known organelles, some of the ones that don't come up as much, but I hope that this really helped, and I will see you next time.